Hi students, so Mr. Cho is at reservist now, so I'll be going through the revision paper 1, okay, and the corrections of this. So I need you to take out your paper and do your corrections. Okay, let's go through. Okay, so question 1 is talking about distillation. So we have covered this before in class. So remember that this part, right, this is called a solution. So this is made up of water and sodium chloride. Okay, so sodium chloride, commonly known as table salt. When you dissolve it in water, you can call it a salt solution. This part here is where the water is going out, and this part here is where the water is going in. Okay, so sometimes it can change, okay, but actually to me it's okay lah. I won't test you on this whether the water goes in or out. Okay, what's more important is you know that this area is called the condenser. Okay, so once the vapor has condensed into a liquid, then it will turn into the distillate. Okay, so distillate is a pure solution. In this case, where we have dissolved sodium chloride with water, right, it's salt water. What we'll get is that the pure water or the distilled water will be collected in this flask, and what will be left behind here will be the salt, right, because it's salt crystals. Okay, so let's answer for question 1 is D. Okay, so what temperature will be shown at the thermometer? It will be 100 degrees Celsius, right? That's because water is evaporating and water has a boiling point of 100 degrees Celsius. And what will be left in the flask will be this white residue, right? The white residue is actually the salt crystal. Okay, question 2. Question 2 says, two liquids N and M were heated at a constant rate and distilled, and the volume of distillate was collected over 14 minutes. So we can see that N, right, starts to evaporate first, and it is being collected first, okay, and then M is at a later time. So that means that M has a higher boiling point, right? It takes a longer time for it to evaporate as compared to the liquid N. So answer is B. Question 4. So forensic scientists can use science, right, to investigate this bloody handprint. So because this bloody handprint can be broken down into soluble components, right, we can use chromatography to separate the components. It's not the best way, right? There still are better ways out there, but if you're talking about chemistry-wise, chromatography is one possible way. Other techniques will include using matching the fingerprint to the database, right? The fingerprint database. Okay, question 5. So talking about the cells, right, tissue, organ system. So if you're looking at a section of a leaf, what we can see is that when we want to see an organ, the organ is the whole leaf, right? So remember that one whole leaf is an organ, and we can see a few cells together, right, make up one tissue. Okay, so it cannot be H, it cannot be I, because H is a cell, I is a cell, but F is a tissue. This answer is C. Question 6, division of labor. Okay, so we didn't really cover this in the G2 science syllabus, but don't worry. Basically what they're asking about is which cell is single cell organism, right? And this Euglena cell, which we have covered before, remember that green little cell with the tail, the flagellum, right? That's called the Euglena. Right? It's a single cell, and so it's single cell, they're not able to divide any work as compared to multicellular organisms like this shark or fly, right? They can actually, different parts of the cell of the insect or the shark can do different things, right? That's called division of labor. Okay, last question. Arnold's family has certain characteristics. So the organelle that controls the characteristics is found in the nucleus, right? So the DNA is found in the nucleus of the cells. In every cell of the body, there's actually DNA, and this controls how a person, how the person has genetics. The person's genetics. Sorry, said wrong. Okay, let's move on to section B. Okay, so in section B, this is a little something from Sec 2 called osmosis. Okay, but for now, you can just copy the answers. Excuse me. So when a sample of red blood cell and plant cell was placed in pure water, the red blood cell bursts. However, the plant cell remained unchanged. So why did the animal cell burst but the plant cell did not burst? That is because the animal cell does not have a cell wall. Okay, but there is a cell wall in the plant cell. Okay, so this plant cell wall, right, helps to prevent it from getting burst. Now the reason why is because of osmosis. So we'll cover this in Sec 2, but to put it briefly, water can enter into the cell and cause it to swell up. And when it swells too much, it will burst. The plant cell can also swell up, but because of that cell wall, it does not burst, right? Because it has something to protect it. Okay, so the cell wall helps to maintain the regular shape of a plant cell. Okay, so for the drawing, right, there's a few important points that you must be aware of. Right, so this is how the teacher will mark your drawing. First of all, your drawing must be clear, it must be neat, and there should not be any shading. That means that you don't need to make it an artwork. This is a scientific drawing, so you are supposed to make it very clean. There must be a correct proportion of the cell structures. So your nucleus should be quite large, right? Your chloroplast should be smaller than the nucleus. Your vacuole should be quite large also. Remember your large central vacuole. And all of them must be labeled correctly. Lastly, make sure that you draw your cell as large as you can, at least 70% of the space provided. Okay, so if there's no box, you just imagine that this whole area, right, the empty white space is your area to draw. Okay, and of course your labeling and spelling must be correct. Okay, let's talk about the organelle that is present in the leaf but absent from the tapioca root. Okay, so in the leaf, you ask yourself, why is the leaf green? That's because the leaf has chloroplast. Okay, and so the chloroplast contains chlorophyll and is able to do photosynthesis. But in the roots, right, it's underground, there is no sunlight, and so there's no need for it to have chloroplast because it cannot do photosynthesis. So that in a sense, the plant is quite smart. It's not going to waste resources making a chloroplast inside the roots. Okay. Lastly, this question about chromatography. Okay, so what does it mean to be pure? Right, so if you look at this, we have 3 perfumes, 1, 2, and 3, and then we have 5 substances, A, B, C, D, E. So for substance C, there's only 1 dot, and this dot indicates that this substance has dissolved in that solution and has moved up a certain distance. And because there's only 1 dot, that likely indicates that it's pure. Okay? So what about E? E is a mixture of 3 different components. Okay? And what component is it made up of? It's made up of perfume 1. As you can see, it's the same look as perfume 1. It is also made up of perfume 2. And it's made up of 1 unknown substance. Right? It could be a perfume or something else. And therefore, you can just write 1 other component. Okay? Or you can write unknown component. That's fine. Lastly, let's talk about why perfume 1 can travel further than other perfumes. So why did perfume 1 go further? Now how chromatography works is that it goes by the solubility. So if something is very soluble, it travels further with the solution. If something is not soluble, it will kind of be left behind, or it will not even move much. So perfume 1 is the most soluble in water. And if you look at D, substance D, right, we're not sure if this is actually something went wrong or it just didn't want to move. Okay, but most likely it's because substance D is not soluble at all, and so it cannot be dissolved in water and it will not move. Okay, so if a student is allergic to A and E, right, the word allergic means that they will have a bad reaction to it, then what should they use? Okay, they should use perfume 3 because perfume 3 does not contain any of the substances that student is allergic to. Okay, let's look at that. Perfume 3, right? Okay, so perfume 3 is this dot over here. The student is allergic to substance A and E, so he cannot have this dot, he cannot have this dot, he cannot have this dot, this dot, this dot. Okay, so the only thing that he must have is this one. And what about B? Can he have this thing? Wait, no, I got confused also. Yeah, he is allergic to A and E. Yeah, so he can take perfume 3. Yeah, that makes sense, right? Okay. Yeah, okay, so that's the end of the corrections. Now you would be given the next revision paper 2. I'd like you to take the time to do it carefully. Use your notes or use your textbook to guide you along the way. I want to see the best quality answers from you. Okay, and then we'll go through this next week. Okay, all the best, and I'll see you next week. Bye bye.